Hello, my name is oluwanifemi Adekanbi Femi, and I will be presenting gut microbiome for class 011 group C. Um, Gut microbiome shows what type of microorganisms are present in the gut of animals and humans, and they are important because it correlates with diseases such as obesity, gastric cancer, IBS/Crohn's disease, autism, allergies, and type 2 diabetes. So our question was, does the age of people affect the gut microbiome? And our hypothesis was the age does affect the gut microbiome, and we determined that because we felt like the older ones will have similar gut microbiomes and the younger ones will have similar gut microbiomes. So we had 4 samples of peoples. We had 2 females and 2 males. We had two older we had a younger female, a older male. We had a younger female and an older female. We had a younger male and an older male. And we chose the samples because they correlated with our partner group choices in terms of gender while we did age. And our group isolated the DNA from the samples and quantified and amplified the bacterial genome, normalized and then analyzed the result from our experiments. And this allowed us to understand the compositions of the gut microbiome for each samples and also to make comparisons. The primers we used were bacteria, fungi, firmicutes, and bacteroids. And based on our results, we determined there was no correlation between the microbiomes of an animal tested who had similar age range. Therefore, our hypothesis was not supported. And we determined that in the future we could alter our experiment by changing the constitution of our dilution to allow a higher amount of bacteria to be present and tested. And we could also test different bacteria and fungal presences, as they could allow us to detect the concentrations and vary in the presence of microbiome to be more accurate. So gut microbiome, as we said before, is important because it detects the bacteria and fungi amounts present in the gut, and they shows the overall health of the person or animals which it was taken from, so they can understand how healthy the person or the animal is. And to expand on the health of the person or animal, understanding the gut microbiome allows us to understand possible health conditions. And the gut microbiome health can be correlated with diseases or ailments that the person or animal has. Examples include gastric cancer, obesity, and Crohn's diseases, and so many more. And by finding out the health of the gut microbiome, we're finding out if the health of the person or animal is good. And once again, our hypothesis was, does the age of people affect the gut microbiome? And our hypothesis was the age does affect the gut microbiome, and it will be shown by the older people having similar gut microbiomes to one another and the younger people having similar gut microbiomes to each other. And then we have 3 references. So for methods, we gathered 4 tubes and we filled each tubes with samples from 4 different peoples, and we added 1,000 microliters of inhibited X to each microcentrifuge, and we put those tubes in centrifuge for 1 to 2 minutes so that we can separate the pellets from the supernatant. And then once we centrifuge them, we place them in a heat plate for 70 degrees for 2 minutes, and then we centrifuge them again for 15 seconds. Then we placed all 4 back into the heat plate, and we left them for 3 minutes instead of 2 minutes. And then once the 3 minutes had passed, we centrifuged all the samples for 1 minute at 14,000 rpm, and then we pipetted 15 microliter of proteinase K into the 4 microfuge tubes, and then we labeled them again 1 through 4 with our group number, and then we pipetted 500 microliter of the supernatant from each of the first 4 tubes into their respective new microcentrifuge with proteinase K. And then once we did that, we pipetted 500 microliters of buffer into the tubes, and we centrifuged for the 4 tubes for 15 seconds and incubated at 70 degrees for 10 minutes. 10 minutes, right? And then once we finished incubating them, we added ethanol to the microcentrifuge tubes. We added 500 microliter of ethanol to the microcentrifuge tubes and vortex them, and we centrifuged these 2 for 30 seconds and added 750 microliters of lysine to the 4 new Chiadin tubes, and then we label them again 1 through 4, and then we close the lid, centrifuge the tubes for 1 minute at 14,000 rpm. Then we removed the tubes and discarded the filtrate, and we did not throw away the plastic tube upper cover. And then we added 500 microliter of the wash buffer into the tubes, and we closed the lid and centrifuge for 1 minute at 14,000 rpm. Then we discarded the filtrate, added 500 microliters of wash buffer AW2 into each of the Kiadin tubes. We centrifuged them for 3 minutes at 14,000 rpm, discarded the filtrate again, and we centrifuged for 3 minutes at 14,000 rpm. And then we added ATE into the 4 new tubes. We added ATE of 200 microliter of buffer ATE into the spin columns of the Kiadin tubes. We put the spin columns into their corresponding microcentrifuge tubes. We incubated for 1 minute at room temperature, which was the vertex, and then we centrifuged them for 1 minute. We removed the spin column and closed the caps of all microcentrifuge tubes after removing the spin column, and then we discarded them. After finding the DNA concentration, we did a 260 to 80 nanometer ratio, and we got for the first one we had 2.06, for the second sample we had 1.91, the third sample we had 2.01, and the fourth sample we had 1.59. The 260-280 nanometer is used to identify the purification of DNA, and when the purification is between 1.5 and 2.2, they are considered to be ideal pure DNA, and all our 4 samples were between 1.5 and 2.2, and then we interpreted all 4 of them to be ideal / pure DNA level for each 4 of the samples. Then we continued and we found the volume of samples in nucleus-free water, and then for our each tube we had the sample / index number to identify which was which, and then we found the volume of sample, and then using that we used that to find the we did C1 times V1 equals C2 times V2, where C1 was constant at 1 microliter and V2 was constant at 100 microliter, and C1 was the DNA concentration level we found using the UV absorbance spectrophotometry. Then we used that to find the volume of water by using the final volume minus volume of sample to give us volume of water. And since, for example, number 1 we did 100 microliter minus 1.4, it gave us 98.61, so that was going to be the final volume of water we were going to get. And so we did that for all 4 of our samples, and our results were for 1 we had 98.61, for 2 we had 98.98, for 3 we had 93.71, and for 4 we had 99.31. And next we did the bacteria and fungi, and we used CQ values and relative abundance analogs, so CQ ratio of bacteria versus fungi detected by the qPCR, and this was our table. So we had our positive, our negative, we had sample 1, 2, 3, and 4, and then we had the bacterial value and the fungal value. Then we used the laptop to find the analogs for both of them, and then the bacteria fungi ratio. Professor Dabrisiagi said not to find the bacteria and fungi analogs for positive and negative, so we left that at that. And I know anything above 25 is considered to be rare or absent, and our positive was below 25 and our negative was above 25, which would have been absent. And for our bacteria sample, all 4 of our bacteria values were considered to be rare or absent because they were all over 25. And for our fungi sample, our positive was and negative were, our positive was below 25 and our negative was above 25. And our first, third, and fourth sample were above 25, so they would have been considered rare or absent, and our second value was below 25, so it was present. And then we found the bacteria / fungi ratio for all of them except the positive and negative once again. And then we did the same thing to our bacteria and firmicutes, which we tested next, or the next class lab. And for our positive and negative, we did the same thing by now calculating the antilogs for the bacteria and the firmicutes and the bacteria firmicutes ratio. And both our bacteria and firmicutes were above 25, so they would have been real absent. And our first sample for bacteria was below 25, while our second, third, and fourth were above 25, which were real absent. And for our firmicutes, our positive was also below 25, and our negative was above 25, which would have been real absent. And then our first sample, second sample, and fourth sample were all above 25, which would have also been real absent. And our third sample was lower than 25, so it was present and it was 18. And that concludes my presentation. Thank you very much.